This video covers the concept of what a species is, so we'll look at different species definitions, and then we'll talk about the mechanisms by which new species form, the process of speciation. Let's first clear up how you write a species name. A species name is represented as a binomial. For example, this bird, the little green bee-eater, is Merops orientalis. The first word of the binomial represents the genus, Merops in this case. The second word of the species name is called the specific epithet, so it modifies which species in this genus. And really you have to have both of these together to fully describe the species. Now, a couple of things to notice about how to write a scientific name correctly is notice I have it here in italics. If most of the text that you're writing in is just regular font, you need to put it in italics if you're typing. If you're writing it and you're, say, writing cursive, the proper way to do that would be to underline both of the words. Not as a single line, but underline, for example, Merops, and then underline orientalis. The other thing to notice is the genus is capitalized, but the specific epithet is not. Now, we use this naming convention using Latinized binomials so that scientists around the world can use a single agreed upon name. Because, of course, there are going to be different local names for different species. For example, this bird is found throughout Southeast Asia, and in different countries it has different common names. So I said it's called the little green bee-eater, but in other areas it has different names in that local language. But if I go to a conference and people know bee-eaters, I can just say Merops orientalis and people will know what I'm talking about. So what is a species, though? I mean, basically what a species is, it's just a specific kind of organism. And we think that a species, that distinction is something that's kind of naturally defining and a fundamental taxonomic unit. Because you realize the other taxonomic units and the traditional classification, Linnaean classification, those are artificial, like a genus. What are the bounds of a genus? How many species go in a genus? That's something that a scientist has to basically determine. And there are some criteria that you can use to say, okay, these species belong in the same genus because they're more similar to each other than other species and other genera. But we think that there's something about a species that is a natural biological unit. And this is because really a species is the smallest evolutionary independent unit that's associated with the potential to interbreed. We're going to talk about interbreeding as an important factor that makes individuals of a species more similar to each other than they are to members of other species. So they have the ability to breed with each other, but they're genetically isolated from other species. And that biological process is what basically sets species boundaries. So it sounds like that should be, if it's a naturally defining thing and we know which can interbreed, seems like defining what a species is should be fairly straightforward and that there should be objective criteria for doing this, but actually it's fairly difficult. One thing that if you want to get two biologists in a disagreement about something, ask them what their preferred species definition is. But the reason for that is actually interesting in itself. There are certain lineages that are in the process of becoming new species. And we'll talk about that later. That's the process of speciation. And they can be at different points along in that process. And different scientists may use different criteria to say, no, there's still one species now. It looks like they are diverging. Others will look at them and say, no, they're diverged enough that we can say now that those are two different species or multiple species from what used to be considered a single species. That's when we can get into some different opinions associated with where those species boundaries are. So the question may differ between different individuals. How much does it take for diverging populations to become two species from one? And what are the criteria that are used to determine whether they are distinct species? Do you need morphological differences? Do you need a set degree of genetic differences? So these are some of the interesting things that scientists may argue about. But in some regards, that's interesting, right? Because that means that's more evidence that evolution is occurring. We actually have different lineages in the process of speciation showing patterns of macroevolution. So I'm going to talk about several species concepts. The first species concept I want to talk about is called the Morphospecies Concept. In this concept, they define a species as the set of organisms that have a specific degree of similarity to each other, but they look different from other sets of organisms. Now, there are some benefits of this definition. It can be used for extinct species. It can be used for asexual species if there's morphological distinctions. But there are also some problems with this definition that some people don't like. In some regards, it lacks a firm philosophical basis. Defining the limits of a species can be subjective. What is defining as a morphological characteristic that's significant? In some cases, it may be bristle numbers on flies that define two different species. Is that significant or not? Well, it kind of depends on if that's linked to genetics and the degree of the genetic differences that may indicate the length of time these two species have been separated. But by just looking at the morphological differences, you're not really getting that. Some organisms you really can't apply this to because they just don't look very different. So if you think about bacteria, there are three basic types of bacteria just morphologically. We have spheres and rods, and then the curlicue bacteria. But we know there are many more species of bacteria than that based upon other criteria, biochemical interactions and DNA differences. But the Morphospecies concept, strictly applied to morphology, is not very useful here. The next I want to talk about is the biological species concept. Here it's defining a group of potentially interbreeding individuals in natural populations that are reproductively isolated from individuals in other such groups. So now we're starting to get to the biology. We're starting to put biological mechanisms that may cause this self-defining characteristics of species that we talked about previously. And this is really the species concept for most biologists. This is the one that most prefer. And it's actually used in the Endangered Species Act, and so it actually has some legal ramifications on conservation issues and regulatory issues. So some of the benefits of this definition, again, it's putting species boundaries in an appropriate biological context associated with the biological processes of population genetics that we've talked about, where different populations can be isolated from each other, where they may have been formerly interbreeding, and after a while, if there's lack of gene flow, they may diverge into different species genetically, behaviorally, morphologically. But the mechanism for this change is the genetic isolation. Now, there are some problems with this definition. You can't apply this to fossils. Comparing different fossils that you find, you could apply a morphospecies concept if they look different to a significant degree. But you can't see if they could breed, right? There's just no way that you can apply the biological species concept here. And if it's an asexual lineage, again, potential that two individuals could breed is just irrelevant. It's a moot point. And one aspect of the definition that some people vary on is the statement that we're dealing with what happens in natural populations. Some people say that you can test the biological species concept by putting organisms in an artificial situation and see if they'll breed. Other people don't like that. But if you are restricting it to the strict definition where you're talking about what can happen in natural populations, how do you deal with situations like this? So this is the distribution of a little bird called the brown-headed nuthatch through the southeastern part of the U.S. But there's also a little population on Grand Bahama Island that was probably established because of a founder event on this island. And if you go to this island, you can clearly identify them as brown-headed nuthatches. They do sound a little different. They do show some slight morphological differences. So you might be able to apply the morphospecies concept to determine if those distinctions are enough to call this island species a new species, or is it just a population still of the brown-headed nuthatch? Well, to test it under the biological species concept, you'd have to see if they could breed. But if they never run across each other because this population was just founded by a few individuals a long time ago and there's not normal gene flow, you just really can't test that. So then we're just left with quantifying the genetic or morphological or behavioral differences to determine or to kind of guess at the likelihood of breeding. Well, that's not probably the best way of doing it, but can we use this information in other ways in another species concept? And that leads us to the phylogenetic species concept. Here, a set of organisms is defined as a species if they have a distinct genetic history from other groups. So this is looking at how species exist through time, which is associated with have they interbred in the past or have they been genetically isolated. So you can use morphological, behavioral, and genetic data to see which sets of populations make up the smallest monophyletic groups or the smallest independent groups with genetic distinctions For example, all of the bees here, these may represent 3 different subpopulations, but they don't show really much genetic distinction. That's just one species. But they may show some pretty significant genetic distinctions from lineages A, C, D, and so forth. So the benefits of this definition is the phylogenies allow us to objectively identify which lineages are genetically isolated. So where gene flow has been broken in the past from formerly interbreeding populations to the formation of new species. And it can be applied to any living organism. You might even be able to say that it could be applied to fossils if you use the morphological distinctions between fossils and you use them to create a phylogeny. So it's kind of a combination of the morphospecies and phylogenetic species concept. Problems with the definition? It takes a lot of time and money to produce phylogenies. Now, this is not nearly as problematic as it used to be in the past, but we don't always have the detailed phylogenies needed for a group of organisms to see where these distinct lineages are existing. So that just means we got a lot of work to do. Some people don't like this definition because when you apply it, it tends to lead to a lot of splitting, meaning what was formerly considered one species, the phylogenetic information may indicate, no, really we have, 2, 4, 6, lots of species. And so it could lead to some confusion in the short term by adding all of these new species and people having to learn them. But I think that's kind of a silly argument. If the data just show that really this should be a certain number of species more than one, that's just what it should be. So let's give some examples of organisms in different lineages that could represent different species and how we could apply each of these species concepts. Here we have a species of copepod. Copepods are incredibly abundant coastal crustaceans, and this species is very widespread. It can be found in Asia, Europe, North American, and if you just look at them, they all kind of look the same from all these populations, and so using the Morphospecies concept, you'd say, yeah, this is just one species. But if you look at the biological species concept, and here I'm taking a little bit of leeway with the potential to interbreed in natural populations, if you take individuals from 38 populations and test them for the potential to mate, you see that you have 8 groups of populations that do show the potential of mating success, but they can't breed across these 8 groups. And then if you use genetic information to try to see where there are distinct lineages, you see that those 8 interbreeding groups are the same ones that are found by the phylogenetic analysis based upon their genetic differences. So the Morphospecies concept says one species, but we do see agreement between the biological species concept and the phylogenetic species concept that there should be 8. What about elephants? Well, traditionally, everybody grows up knowing that there are African elephants and Asian elephants. But the Morphospecies concept actually suggests at least 3. If you look in Africa in different habitats, you have African savanna elephants and forest elephants, and they do show some morphological distinctions. Well, what would the biological species concept say about this? Well, it's not really clear because the 2 African groups are not sympatric. They're what we call allopatric. They live in completely different habitats and they never run across each other. And it's not like copepods where you can just collect some of them and put them in a petri dish and see if they, or in a tank and see if they'll breed, right? You just can't do that with elephants. So it's unclear currently whether they potentially could breed. And again, the traditional purist would say that that's if they don't ever meet up in natural populations anyway, then you just can't address that. Well, the phylogenetic species concept, if you look at genetic distinctions, you see that yes, there are 3 species in agreement with the Morphospecies concept. So again, we have 2 species definitions that are in agreement and one that in this case is not disagreeing, but it's just not really able to inform on the question. And so this has some conservation implications. If you're trying to save elephant populations and you have limited amount of money and resources, and you just spend all of your money on savanna elephants and you let populations of the African forest elephants dwindle, you're losing that genetic diversity. You could potentially be losing a species if you didn't recognize that all African elephants weren't the same. So I started this discussion by saying that species are natural groupings. So is a species the only natural taxonomic unit, or is it natural? Well, in most cases, the species defined by one concept are also the species defined by another. I mean, if you go outside right now, if I started showing you different birds, I'd say, okay, that's a red-eyed vireo, that's a white-eyed vireo, that's a yellow-throated vireo, there's a northern cardinal, there's a mockingbird. And you'd look at them and say, yeah, they look different, they sound different. And if you started looking at nests, you'd see that they all nest differently and don't interbreed. So all of the potential tools, and if you looked at their DNA, you could see it was different. All of those are in agreement about those species boundaries. It's only, again, in those cases where there is recent divergence from a common ancestor that make it difficult to determine, well, yeah, they're showing some genetic distinctions, but they don't look different yet. So you might find that there's a morphospecies concept of one, but a phylogenetic species concept that suggests more lineages. So I would say that most species recognized by taxonomists, scientists that work on species names, fit also folk taxonomies. You don't have to be a trained scientist, again, to go outside and say, well, a mockingbird is a different kind of animal from a northern cardinal. And there was a famous study by Ernst Mayr where he was cataloging these bird species in New Guinea. And he found out that this New Guinea tribe had 174 vertebrate species, so more than just birds, just vertebrates in general, that they recognize as different kinds of animals, so basically species. And then really this differed only by 4 in what Ernst Mayr was recognizing. He recognized 178. So he was maybe recognizing some very subtle differences that just weren't important from their point of view. So I think that's a pretty good test that shows that yes, species boundaries oftentimes are naturally forming and fairly easy to see. But it is really interesting to see those cases in which speciation is occurring and why that causes arguments about how many species may exist in those situations. So it may just be hard to define exactly what those boundaries are in all situations, but that's just, again, proof that evolution is occurring. So let's talk about the process of the formation of new species, that is speciation. The key mechanism associated with speciation is genetic isolation. You have to get genetic isolation between groups of individuals that were formerly interbreeding into 2 or more groups. And once they become genetically isolated, they'll slowly start to accumulate genetic and morphological and even behavioral differences in each of those new lineages that are going to form new species. So I'm going to talk about 2 broad classes of how speciation can occur based upon geography. If we have a species that existed in one space at one time, but somehow eventually becomes separated into 2 geographically separated populations, we say that those now become allopatric. Allo means different, patric means country, so now there are 2 geographically isolated populations of what was formerly a species that was all potentially interbreeding in the same location. If that separation exists through a long enough time period, again, you're going to start to accumulate differences. If somehow the environment changes or the circumstances changes where individuals in each of these new species come back together and they can't interbreed, they're not going to be exchanging genes, they're going to keep their genetic, behavioral, and morphological distinctions, then speciation has been completed. But the mechanism that caused that differentiation was the time period in isolation. The allopatry was important. But you can get allopatry in 2 basic situations. So we could have a population that starts off on a mainland, and a nearby island may receive a few colonists through kind of a one-time gene flow establishment of this new population. But there's not really subsequent genetic exchange between them. That's a form of allopatric speciation through dispersal. So the example of the brown-headed nuthatch I gave earlier would be an example of that. And several lectures back, I was talking about scrub jays. Many of the scrub jay situations, for example, going from California to Santa Cruz Island, that would also be a dispersal form of allopatric speciation. In other cases, the organisms themselves may not change, but habitat changes around them so that maybe we have a situation where this is all forested habitat and it's over a large geographic range, but rain patterns change so that this area becomes much drier. We still have the preservation of forest here, but now we have a grassland in here. and that is a habitat that may not be appropriate for what was formerly one species. But now this forest patch and this forest patch, the individuals in these separated populations again begin to accumulate these differences and form two new species. So it's still because of separation through allopatry, but these individuals didn't move, they just got isolated because of this geographic change. We call this vicariance. Here's an example of allopatric speciation by dispersal. If you go to the Hawaiian Islands, the Drosophila there, the genus has so many species and it's amazingly diverse. Here's just an example of some of that diversity. And if you collect flies on the different islands and you do their phylogeny and compare it to the history of how these islands form, it shows a clear pattern of allopatric speciation by dispersal. So the bigger islands here, the main island of Hawaii, is the youngest island. Then you get over to Maui, Molokai, Oahu, Kauai. As you go this direction, this is a chain of islands that are formed through volcanoes that erupted much longer ago. So this is associated with building this island through more recent volcanic activity. Well, if you collect the flies on the different islands, you see that the older lineages are associated with the older islands. And the more recent species, these are associated with the newer island. So as this island formed, you had individuals from this island hop over here to establish a new population. But before that had happened, we had individuals from a species on Molokai disperse and form a new population here on Maui. So it's kind of this leapfrog from older islands and establishing new populations initially that become new species as you get new islands. And there's even geographic isolation within the big island of Hawaii that's led to separation of species just on the one island, on the Hilo side and the Kona side. Here's an example looking at snapping shrimp. And if you look at the geological history of North and South America, we know that the connection between North and South America associated with what's called the Isthmus of Panama land bridge used to not be there. And so there was connectivity between marine species in the Caribbean and the Northern Pacific because it was a complete access through these marine environments. But once that land bridge was established because of sea level changes, now there's a barrier that's popped up. Now these organisms didn't necessarily change. They stayed on their individual sides. The establishment of this land bridge separated populations in the Caribbean and in the Pacific that eventually formed new species. And this happened about 3 million years before present. That's what MYBP stands for. And if you collect individuals in different species in the Pacific side and the Caribbean side, you see that they have this sister species relationship typically. And that's what would make sense if that land bridge, that vicariant event, separated a Pacific population from a Caribbean population that was formerly interbreeding. So they have a common ancestor, but when the land bridge came up, you formed from this one species, Pacific copy and a Caribbean copy, and then over here we again had a Pacific and a Caribbean, and you just see that throughout this phylogeny. When you look at a phylogeny and you see that there are two species from a common ancestor, we call these most recent lineages from a common ancestor sister species or sister lineages. And in laboratory studies, these sister species will still attempt to mate with each other. They don't with more distantly related species. They attempt to mate, but they have acquired enough genetic distinctions that they can no longer produce fertile clutches. And this is going to be an important point that we're going to talk about later with the potential if two species happen to come together, say if the land bridge disappeared and they had access to each other again, would we still maintain those species boundaries? In this case, no. So it appears that in most cases, there's data to suggest that a period of isolation, geographic isolation, is really the most important way that new species form, either through dispersal or vicariant. But that allopatric speciation is fairly typical. Can you get genetic isolation without geographic isolation? So if we have a group of organisms that occupy one area, they divide themselves up into two groups that become genetically isolated. Can that happen? We call that sympatric speciation. Sym meaning same, patric again, country or land. So we're getting genetic isolation without any geographic distinction. They're in the same land. Well, this could happen through polyploidization. We talked about this earlier in the semester, because polyploidization usually results in instant reproductive isolation, because you can't have polyploids mating with diploid individuals because their offspring would be triploid, and that causes all kinds of genetic problems. So it generally leads to sterility. And this again may be very important for many of the land plants that have polyploid origins, and leading to the formation of polyploid species that become separate from their diploid ancestors. It's also seen in some lizards and some fish, but it's more common, as I mentioned, in angiosperm plants, especially when they have selfing capabilities. Other chromosome changes could have similar effects. So if you have a merger, say, of two chromosomes into one, then you're changing the chromosome numbers again, and that could lead to the inability of individuals that don't have that mutation to mate with individuals that do. Now, I don't have a slide for it, but there's another way that you could get sympatric speciation. We talked about it earlier. If you have extreme disruptive selection, where individuals with different genotypes only mate with individuals like them, and that's strict enough, that can lead to genetic isolation in the same general area. So you may go back and look at your notes on disruptive selection to revisit that issue. But if that was persistent enough for a long enough period of time, it could lead to the formation of new species sympatrically. So we may get genetic isolation between different lineages through allopatric or some sympatric mechanisms, but what happens then? First, we can have genetic divergence between these isolated groups through genetic drift. This is going to be particularly important in situations where there's allopatric speciation through dispersal, where you may have a few colonists establish a new population. Just that small number of individuals is going to lead to some genetic differences pretty quickly between the founding colonists and the ancestral species from which they come. Same thing could happen on, say, the very edge of a habitat that may be, say, a forested habitat. If a little island of that habitat remains and it's isolated through a vicariant event, again, that small little isolated satellite population is probably going to have few individuals and be under extreme genetic drift pressure that could lead to genetic differences between the small population and the big population. But when we have species that are isolated into different areas, those areas may have different natural selection patterns. And so here's several examples of studies of insects, of vertebrates, including fish and birds, in which there have been different selection pressures associated with different genetically isolated groups. So it could be associated with switching between host plants. That's a really common thing in insects, as you can see. Using different microhabitats for foraging, benthic versus limnitic forms of fish, that causes them to become isolated, and selection for different genes for those different foraging habitats. So a variety of things can happen that lead natural selection to produce adaptations differently in the different populations that are isolated. And this is just going to lead to them becoming more and more genetically distinct. Sometimes isolated lineages can develop different sexual selection patterns. Perhaps females change female choice preferences, or males use different male-male competition traits in different populations when they're competing for females. So for example, a female bird may switch from preferences for long tails to a preference for brighter feathers. These new sexually selected traits, again, are going to drive genetic distinctions between the different populations. Here's an example of how different species of Drosophila can differentiate from each other due to different male-male competition traits. In some species, they act more like some mammals that push each other back and forth. Think about cows or elk and deer. Others, they stand up on their hind legs and they use their front pair of appendages to box each other. But these isolated lineages have developed these different male-male competition traits due to the period of isolation. And again, it's just going to lead to them becoming more and more distinct from each other as they get these new traits. And again, here are just some examples of studies that have documented how this changes. So courtship songs can differ in crickets and different populations that are becoming new species. Sexually selected traits such as wing pattern and coloration, again, song, body size, coloration. So these are fairly common traits in a variety of organisms, both vertebrate and invertebrate. So we've talked about how you get genetic isolation, and during the period of genetic isolation, you may get genetic distinctions come about. But what happens if these two potentially new species come into secondary contact so that the genetic isolation has the potential to dissolve? So if we have two lineages that started off diverging due to allopatry, but then they become sympatric again, then the question is, can they still mate? And if they do, if they don't, no big deal, right? Then you've clearly established that they're new species. If they can still mate, then we have to look at the fate of their hybrid individuals. Are they sterile or are they maladaptive in some ways? If that's the case, if it's significant enough, then you're still probably going to be maintaining those genetic distinctions that will keep them as separate species. But the answers to these questions really are going to determine whether we have formed new species and speciation has been completed, or if these populations are going to merge back together. And some of the mechanisms that we talked about during the genetic isolation, like having different natural selection and sexual selection patterns, may make it where the hybrids just aren't going to be very good. They're going to have less fitness, even if they are not sterile. If that's the case, we should see the evolution of what we call reinforcement. Reinforcement mechanisms help to prevent individuals from the two new species from forming hybrids, and this can help maintain that genetic isolation, even if the lineages have merged back geographically. So you're not relying anymore on allopatry to keep the new species from interbreeding and producing these hybrids. The reinforcement mechanisms help to actually make these less fit hybrids to not be formed. So let's talk about types of reinforcement mechanisms. Again, things that would keep individuals from mating. So we call these pre-mating isolating mechanisms or pre-zygotic isolating mechanisms. So the first may be because they have different ecological or habitat preferences, selection for doing things differently. Different host plants, for example, different microhabitats. So an insect, one species may exist in the leaf litter, while others exist on a tree. Well, that in itself may prevent them from running across each other. They may be in the same forest, but if they never run across each other, they're not going to have the potential to mate and form these maladaptive hybrids. So that would be a good pre-mating isolating mechanism. And there should be selection for them sticking with their own habitat, because again, that's going to prevent them from mating with individuals that wouldn't produce good offspring. Or maybe they're exactly using the same ecological setting and they run across each other on a daily basis, but if they evolve reproducing in different seasons, so some breed early summer, some breed late summer, that also would be a mechanism that natural selection would support if it kept individuals of the two species from interbreeding and forming these bad hybrids. And we've already talked about how you can have different sexual selection, female choice traits, and male-male competition traits. That would be an example of a sexual or behavioral isolation, again, that would prevent the formation of hybrids. Mechanical isolation, this is a situation in which they may be in the same area, they breed in the same season, they have the same close enough sexual selection behavior so that they attempt to mate, but if their genitalia just won't fit together, that would lead to mechanical isolation and prevent the male from successfully fertilizing the eggs in the female of the other species. Sometimes this is referred to as the lock and key hypothesis. It's been studied in insects primarily. Pollinator isolation, this is one that's restricted to angiosperm plants. If they have different flower morphologies that attract different pollinators that are less likely to visit the individuals of the other species, again, that's going to, if you keep your pollen just to your own group, that will prevent hybrids from forming. Gametic isolation is if none of these things work. If the males and females mate, but their gametes just are not chemically compatible and there can't be successful fertilization, you're not going to form a zygote. So reinforcement mechanisms should be selected for if they can efficiently prevent individuals from maladaptively forming hybrids that just aren't going to be good. But you can still maintain genetic distinctions in kind of less efficient ways. We call these post-mating or zygotic isolating mechanisms. So you may form hybrids, but if they're inviable, if they die during development, again, this is going to keep the species from merging back together again. Or if the hybrids are sterile, they may exist for a period of time, but if they can't transition their genes from one species to the other, Again, you're going to maintain that genetic distinction. Hybrid breakdown is where you may get a little bit of gene flow in one generation, but the next generation fails. That would also maintain some genetic distinctions. So these post-mating or zygotic isolating mechanisms aren't as efficient as the reinforcement mechanisms, the pre-mating or pre-zygotic isolating mechanisms, but it may start off with these, keeping the species separate, but if you evolve the reinforcement mechanisms, that would be highly preferred. It would be better for all the individuals involved to not waste time with the production of hybrids that just aren't going to work. But it turns out in some cases after secondary contact, hybrids may have equal fitness to that of their parent lineages. Now, if this is the case, then the lineage distinctions, the two what we call incipient species that were about to form, are just going to merge back into one species. So you may have started off with forming two species, but if they just are not isolated genetically, once they come back together again, they're just going to blend back into one. There's actually a third possibility. If, however, the hybrid have really good fitness, maybe even better than that of their parents, in the hybrid zone, then we go from one species ancestrally that may have split to form two species, but the hybrids then may make a third species. So I'm going to give you an example of that. Let's say that we've got a group of organisms that the blue species over here has the highest fitness. It's adapted to the ecological setting over here, but there's a clinal variation. So there's a gradual change in the ecosystem so that this area where they overlap has some of the characteristics that the blue species are adapted to and some of the characteristics that the red species are adapted to. But the red species, actually individuals do better over here. But in the hybrid zone, where they can interbreed and they pass on some of the blue genes and some of the red genes, now these individuals that are hybrids may have better adaptations for this intermediate habitat. In which case, you might then form reinforcement mechanisms so that the hybrids just mate with each other. And then you would form three species. So in a large geographic area where you see changes in habitats at ecotones, where two ecological communities meet and have some characteristics of both of the habitats on either side of them, this is where you're most likely to see the formation of these hybrid species. Here's an example of this, looking at lowland sagebrush and higher elevation sagebrush. So in the basin, we see that plants in the basin habitat do the best there. They have the best fitness if they are artificially placed with the intermediate species and the mountain individuals in the mountain species. They can live there, but they don't have nearly as high fitness in that lower elevation habitat compared to the plants that have evolved adaptations to that lowland habitat. Jump over here to the mountain ones. Yeah, the mountain ones have adaptations for higher elevation. They have higher fitness in competition with the other lineages. But if you look at the hybrids actually do better in that intermediate foothill-like habitat. And it's typically associated with body size as one of the key adaptive characteristics linked to different elevations. So we've been talking about phylogenies a lot this semester, and the next video is going to talk about the process of how we actually make these phylogenetic trees, how we collect data on organisms to figure out their degree of relatedness. So that is called phylogenetics.